Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics. This is microbiology, bacteria, viruses and infection. The first part is about bacteria and viruses. So beginning with bacteria, we have a structure of a bacterium. It has a flagellum. This is going to enable it to move or for movement. It has the pili, which are thread-like projections. These help it to adhere onto surfaces. There is a slime capsule. There is a mesosome. We can see here the internal extension. And some bacteria do have plasmids. These are circular strands of DNA. All bacteria have a cell wall, which is made out of peptidoglycan. They also have a surface cell membrane that is made out of phospholipids. They have a nucleoid, which is a single circular strand of DNA. Viruses are very small. They're just arrangements of material as well as protein, and they invade cells in the body where biochemical reactions take place. They can exist and reproduce as parasites only and in living cells. Viruses have a protein coat, and this protein coat is made up of repeating proteins. These are called capsomeres. Viruses do have nucleic acids. This can be DNA or RNA. And viruses do have surface molecules, which are antigens. These are going to be specific to the molecules on the cells of the organisms they infect. They can also have a lipid envelope, and this makes it easier for the viral components to be passed from one cell to the next. But remember, not all viruses have a lipid envelope. Viruses can be classified into DNA as well as RNA viruses. DNA viruses include adenoviruses like of colds, bacteriophages, and so on. They have DNA as their genetic material. We can see it's going to be in there as their genetic material. RNA viruses, these include the tobacco mosaic virus, Ebola, and so on. They have RNA as their genetic material, and they are much more likely to mutate. They do not form DNA as any part of their life cycle. There are other viruses called retroviruses that we're going to talk about later. These include the HIV virus. Retroviruses are a special type of RNA viruses. They have a lipid envelope as well as a protein capsid. They have single-stranded RNA, which controls the synthesis of reverse transcriptase. They contain enzymes, reverse transcriptase, as well as integrase, and these are going to be used in incorporating the viral genetic material into the host cell's genetic material. Going to topic 6A2, how viruses reproduce. The viral life cycle can go through two pathways. There is the lytic pathway as well as the lysogenic pathway. Most DNA viruses do not cause disease immediately, so they will go through the lysogenic pathway. There is going to be repressor genes that make translation impossible. The virus is going to be said to be latent or in a period of lysogeny. However, if conditions are altered, then the lytic pathway can be triggered. During the lytic pathway, the cells are going to burst out, releasing large numbers of the new viral particles, and these are going to invade other cells. And the virus is now going to be disease-causing because many cells are going to be destroyed. Moving on to the life cycle of retroviruses. Retroviruses attack animal cells. We can see there is an attack. The virus is coming in, and the genetic material is going to be released. This is RNA released into the host cell. This is going to be converted into viral DNA using reverse transcriptase. And this viral DNA is going to be incorporated into the DNA using the enzyme integrase. And afterwards, new viral particles are going to be assembled, and these are going to leave the cell by exocytosis. But the viral DNA is going to remain in the nucleus for repeat cycles of making multiple viral particles. Moving on to viruses and disease. There are recognition markers on the cells that are infected by viruses, and these viruses bind onto those specific molecules as a way of attachment in order to enter the cell. Presence of these biological markers determines if a cell is going to be infected by a specific virus or not. And sometimes infected cells are going to produce some enzymes, and these enzymes can lead to the destruction of the cell. Sometimes they release toxins that are going to inhibit metabolism, and the cell is going to be destroyed. The next is 6A3, the growth of bacterial colonies. Bacteria reproduces through fusion, whereby one cell divides into two and so on. When bacteria reach a specific size, replication can begin to occur. So basically, when one cell grows and increases in size, then the cell can begin to divide. And during the replication process, the DNA can be held in position by the mesosome, which we saw previously. Generation time is a time between two cell divisions. And here we have an example of a graph showing the bacterial growth. This is a growth curve. For this one here, we have the number of bacteria, and then we have the log of the number of bacteria. This is more acceptable because it can show you a linear or a relationship that is more easier to understand in comparison to this one, because for this we can see almost there is 0 bacteria. At 1 hour, almost to the end of 8 hours, we still have 0 bacteria. But when we use the log of the numbers, we can see there is an increase that can be visible. Next is the exponential growth rate. This is at the exponential phase. They will ask you questions about calculation of this constant. If a growth curve is given, you get the log number of bacteria at a specific time minus the log number of bacteria at another time, which is going to be the initial time, divide by the log to base 10 of 2 times the time the experiment has been carried out, or the time difference between t and 0. It's used to determine the number of bacteria in a colony, and the formula is NT is equal to NO times 2 power KT, where NT is the number of organisms at time t, and or number of organisms at time is equal to 0, K is going to be the growth rate constant, and T is going to be the time the colony has been growing. Moving on to the bacterial growth curve. This growth curve has different phases. There is the lag phase. This is where the bacteria are adapting to the new environment and getting used to the conditions of temperature and so on. So growth is going to be slowed. And then there is the exponential phase or the logarithm phase. In this phase, there is going to be maximum growth because the bacteria are used to the conditions, there is nutrients and so on. So basically the conditions are going to be optimum. Then we go to the stationary phase. During this phase, the number of bacteria that are dying and the number of bacteria that are being reproduced or that are being formed is the same. So we can say the total growth rate is going to be 0. And as more nutrients are used up, the death phase or the decline phase is going to be reached. Here, reproduction almost stops due to toxicity and lack of nutrients. So we can see there is more bacteria dying, and so the curve is going to level down. Moving on to topic 6A4, microbial techniques. So here we're going to be looking at aseptic techniques. I use this presentation for my students, so I put some YouTube videos. These are links where you can watch some of these videos to learn about the culture techniques. In culturing bacteria, you have to decide on the microorganism that you're going to use. You have to determine the right nutrients, like a carbon source, a source of nitrogen, as well as mineral requirements within the medium. And during the culture, are you going to use a solid, like or nutrient agar, or is it going to be liquid, like in nutrient broth? During the culture, as well as the preparation, the media has to be kept under aseptic conditions. These aseptic conditions include flaming, like you can see here, the flaming of the loop, wiping of the space with alcohol, as well as wearing gloves and working with sterilized equipment. In growing a pure culture, you usually isolate the desired microorganism from others by changing the growth conditions to ensure that only the desired microorganism is going to grow. These conditions could be aerobic or anaerobic. Varying the growth medium can favor a certain strain. For example, put antibiotics as well as antifungals so that one bacteria that has the ability to withstand the antibiotics is going to be able to grow while the others are going to die. That is a form of selection. Sometimes they use indicator media that causes certain bacteria types to change color, and those that do not change color are going to be isolated and they're going to be cultured. Moving on to topic 6A5, measuring the growth of bacterial cultures. So here we're going to look at hemocytometer, optical density, and many more. When they use a hemocytometer, we can see this is the hemocytometer. We can pour 0.1 millimeter cubed of the solution containing the bacteria. And then there are going to be some grid lines within, as we can see here. This is going to be how it looks like under a microscope. The diluted sample of the nutrient broth is going to be placed into this space here. A dye is going to be added. This is to stain the cells so that we can be able to identify those that are dead. It ensures that only the living cells are going to be counted. So you can make a decision either to count the cells that, for example, we can say we're not going to count the cells that are touching onto the edges this side, but we'll count those this side, in that site. And when you make that decision, you have to be consistent for all the areas that you're going to count in. To find out the total number of bacteria in a specific medium, that hemocytometer is calibrated so that, because you remember we put 0.1 millimeter cubed, in the 0.1 millimeter cubed, we're going to count the number of cells. For example, if you have 16 cells, the total number of cells in 1 centimeters cubed is going to be 16 times 10 power 4 due to this. Then you can scale up in order to find the total number of cells in the total solution that you had initially. The next method is turbidity. In this case, we are using a colorimeter because remember, as bacteria grow, there is going to be an increase in the turbidity and more light is going to be absorbed, so less light is going to be transmitted. In using a colorimeter, we usually put a blank. This is a blank. This is the solution of the media without any microorganisms or without any bacteria. And a light is going to be passed through it. This light is going to be of a specific wavelength. And then a detector is going to detect how much light is going to be transmitted through. This is to zero the colorimeter. And then we'll place the sample. This is the sample of the culture containing the microorganisms. Because there is more turbidity, the light is going to be absorbed and we can see less light is going to be transmitted through to the detector. We could use this accompanied with a hemocytometer. Remember, the hemocytometer can estimate the number of cells to draw a calibration curve. And later on, we can use that calibration curve to find the total number of bacteria at any point during growth, because the calibration curve linking the absorbance and the concentration on number of bacteria due to the data from the hemocytometer and turbidity will be already drawn, and that is going to help us to find the actual number of bacteria without needing to go back to use the hemocytometer. The next one is dilution plating. In dilution plating, as we see here, we're going to use serial dilution. There is going to be a sample. So let's say if we have 10 centimeters cubed here, we're going to take off 1 centimeter and add it to 9 centimeters cubed of water. Then this is going to be mixed and a sample of this is going to be cultured on the petri dish. Then we'll transfer 1 centimeters cubed to another 9 centimeters cubed of distilled water. We'll mix properly, take off a sample, and then we can culture that. And then transfer 1 to the next 9, mix, take off a sample culture, and do the same. Culture various in series until we can obtain distinguishable colonies. Now, in the end, each colony is going to be counted as one individual cell. So here we can see in the original concentration, we had 3 cells per that specific volume. So this could be 3 times the number of dilutions. So we can see 1, 2, 3, 4, 5. It could be times 10 power 5. And we can be able to use this to find the concentration of the bacteria in a specific sample by scaling up. In dilution plating, we only consider live cells or viable cells because those are the cells that were able to create distinguishable colonies. But in the turbidity method, we are looking at absorbance or transmittance. Cells that are dead are also going to show some absorbance, so there is no distinguishing between cells that are alive and those that are not. The next is about the area and mass of fungi. This can also be used to quantify growth. In this case, we will measure the diameter of the individual mycelium. This can be used to compare growth because those that grow faster are going to have wider diameter, and those that grow slower, the diameter is going to be small. And this can also be used to study the effect of growth conditions on the rate of growth. For example, we could alter the temperature and then see how the masses or the area is going to be. So the optimum growth conditions are going to give the greatest or wider diameter or wider mean diameter. This should be petri dish, the E should go away. Sometimes they can also measure the dry mass of the microorganisms. This could be done by culturing in liquid medium, removing the sample at various intervals. You could centrifuge to obtain the sample. Then you could dry the sample to constant mass. This could be overnight in an oven. And this is going to give us the dry mass per specific volume of the culture medium. So we can compare different conditions in order to see what gives more mass that is going to have the greatest growth rate. Going on to 6A6, invading the body. Here we're going to look at methods of spread, which include vectors. Vectors are organisms that transmit infections, and these include insects. They can cause diseases like malaria, yellow fever, and so on. The next one is inhalation. Here somebody could cough, they could sneeze, or even just talk, and droplets are going to come from the infected person into the person who is not infected. They're going to get diseased, and this includes measles, tuberculosis, and so on. The next one is ingestion, meaning things are going to be consumed. You could eat contaminated food or food that is not properly cooked. Sometimes this could be raw food and so on. The microorganisms are going to enter into the digestive system, and these are going to cause diseases within the digestive tract. These include salmonella poisoning as well as hepatitis A, which can affect the liver. The next one is vomitives, for example, towels and beddings as well as books, money, and so on. So any surfaces that an infected person could have touched can be a form of contact in order to transmit disease. Then next we go to direct contact. This could be sexual transmission of diseases. These include gonorrhea, syphilis, and so on. This one needs direct contact with the person who is infected. And then there is inoculation. This is where there is going to be breaking of the skin, and inoculation is going to occur in the body. This could be through medically contaminated instruments like shared needles. So the diseases include HIV, rabies, tetanus, hepatitis B, and so on. Moving on to epithelial defenses. The first one is the skin. Our skin contains keratin, which is a fibrous protein, and this is going to be a good physical barrier. There is also sebum, which has antimicrobial properties. We also have skin flora. These are microorganisms whose purpose is to outcompete pathogens for space and nutrients in order to prevent us from infection. We also have surfaces of internal tubes and ducts, for example, the nose, the respiratory system, the urinary system, and so on. They contain mucus-secreting cells, and these contain lysozymes that can break down microorganisms and render them ineffective. Some cells are also going to be ciliated. These are able to move the mucus that contains the dead microorganisms away from the areas of the body. We also have white blood cells that engulf and digest the pathogens on the epithelial surfaces. The next defense, we have the gut. Of course, this contains enzymes that break down the bacteria. It contains hydrochloric acid that provides the pH that most pathogens or most bacteria cannot thrive in. We also have the natural flora within the gut that competes for space and nutrients with any pathogens. And also our body can go through a ring of vomiting in order to expel any ingested organism. Moving on to how bacteria cause disease, we're going to look at endotoxins, exotoxins, and host tissue invasion. Endotoxins, these are lipopolysaccharides. They are found on the cell walls of gram-negative bacteria. The lipid part of this lipopolysaccharide is going to act as a toxin, and it triggers immune response. Usually they have their effects around the bacterial infection site. We can see the endotoxins here. Endotoxins are not usually fatal, but they cause side effects like diarrhea, vomiting, and if these are not taken care of, for example, to replace the fluids that the body has lost, then the person could actually die. Sometimes endotoxins are going to be released because of antibiotics that destroy the bacterial cells, and when lysis occurs, then there is going to be further release of endotoxins. Moving on to exotoxins, these are soluble proteins. They are released by bacteria. These can be produced by both gram-negative and gram-positive bacteria. They have greater effects than endotoxins. For example, they can cause internal bleeding, they can cause damage to cell membranes, they can compete with neurotransmitters, and sometimes they directly poison cells. So this can lead to a decrease in the number of cells, and sometimes it can lead to damage in organs. The next is host tissue invasion, like we see in tuberculosis. Most times this is going to be due to the toxins that are released. Tissues are going to be destroyed and organ damage could occur, as we can see here in these lungs of a person who has tuberculosis. Moving on to case studies, we're going to begin with 1, tuberculosis. Tuberculosis is caused by Mycobacterium tuberculosis, and this could be from drinking contaminated milk or those who work closely related to cattle. However, it can be transmitted from one person to another through water droplets as one person talks or breathes or coughs or sneezes. Then the droplets can be transferred to another person, and that can cause infection. Tuberculosis is more severe among people who have a compromised immune system. When TB infects the body, there is a latent phase and there is an active phase. In the latent phase, usually they form a tubercle and they hide away from the immune system. But when there is a compromise in the immune system, then we can go to the active phase where tissues are going to be infected and there is a lot of destruction that could occur. So TB is going to cause a lot of lung damage, and it suppresses the immune system, making the person not able to fight disease. The symptoms include weakness, coughing out of blood, and this usually appears towards the end of the disease progression when people cough out blood. Like I said already, TB has two stages. There is primary infection. During this period, the person is going to inhale the bacteria into the lungs, and these are going to slowly multiply. If a person is healthy, their immune system is going to try to fight. There is going to be localized inflammation, and this is going to cause formation of MRSAs, which are called tuberculosis. After about 8 weeks, the immune system is going to control the bacteria, and the inflammation could even disappear. Most people have actually come into contact with tuberculosis, but because their immune systems are not that compromised, they do not show any symptoms of disease. However, when the immune system is compromised, then tuberculosis is going to be expressed and the person is going to go into the disease phase. Lung tissues are going to be destroyed and the person could actually die. The symptoms of active TB include fevers, night sweats, there is loss of appetite, loss of weight, and so on. And tuberculosis can be tested using sputum, and sometimes they do X-ray to see if somebody has ever been infected with tuberculosis. So in the exam, expect a lot of case studies on tuberculosis. Actually, they ask a lot about the symptoms. In severe cases, there is going to be a lot of tissue damage. The people are going to lack energy, or the patient. Lung tissue is going to be destroyed. There is going to be coughing out of blood. And actually, these can lead to severe issues with the lungs that could cause death. Treatment and control of TB. TB is treated by using different antibiotics. So during the treatment, this can be done using antibiotics for months, usually 6 to 18 months, and they use different combinations of the antibiotics. To control tuberculosis, you need to avoid crowded areas, you need to take pasteurized milk, you need to go for vaccination or immunization, and you can improve standard of living. In topic 68, case studies of disease, and this is HIV. HIV is a virus. This virus attacks the T cells of the immune system, making the body prone to opportunistic pathogens like TB, pneumonia, as well as more. If a person has the HIV virus, we say they are HIV positive, and this can be tested by finding HIV antigens in the body. The disease is called AIDS. AIDS is called acquired immunodeficiency syndrome. People who have AIDS have symptoms like fevers, persistent diarrhea, weight loss, and so many more. HIV can be transmitted through exchange of bodily fluids. It could be through inoculation with infected blood. It could be between mother and fetus during birth or breastfeeding. And HIV is a retrovirus, like we saw already. It attaches onto the T helper cells and infects them. After entry, it controls the host DNA and replicates, creating many viral particles. As the viral particles leave, they destroy the T helper cells, and this is negatively going to affect the immune system, since T helper cells are involved in specific immune response, like stimulating of the B cells as well as the T killer cells. So the body is going to be vulnerable to infection by pathogens because the immune system can't effectively fight infection. So like we said already, opportunistic pathogens like tuberculosis are going to take over, and this could lead to AIDS, which is the disease. The HIV virus attaches on the surface of the cell, and then it's going to release the genetic material into the cell. The genetic material is going to go through reverse transcription by reverse transcriptase enzyme to produce DNA. We can see that here. And then the DNA is going to be taken into the nucleus by integrase and incorporated into the host cell DNA. And all cells that are going to divide from that cell will contain the same DNA. And afterwards, a lot of viral particles are going to be produced from this cell as the cell breaks down to release the viral particles. The course of HIV and AIDS infection is in 4 stages. The first stage, this could occur in the first few weeks after infection. In this case, the person is HIV positive. Symptoms could be some headaches, tiredness, swollen glands, and so on. However, this could disappear depending on the person's immune system. So this person goes into stage 2, which is the asymptomatic stage or asymptomatic phase. This could vary For some people, it's years. During this phase, viral replication and T helper cell infection occurs, and T killer cells can destroy the infected cells, decreasing the rate of spread. So it could take longer or shorter. And when the person transitions into stage 3, this is going to be the symptomatic phase where we say the person has AIDS. The viral load is going to increase and the immune system is going to weaken because many T helper cells are going to be destroyed by the T killer cells. If the cell count decreases to about 500 to 200 per centimeter cubed of blood, then the person has AIDS. During this phase, people will have fatigue, weight loss, diarrhea, night sweats, thrush, and so on. And then towards the end of their life, the person is going to go into advanced AIDS. In this case, the T helper cells are going to decrease severely. There are going to be severe symptoms. There is going to be excessive weight loss, cancers, TB, meningitis, dementia, and so on. And this is eventually going to lead to death. HIV can be prevented by education to improve awareness. We could do more testing so that people can know their HIV status. People could practice celibacy, meaning not involving in any sexual intercourse, or they could use condoms. You could also do that by having one sexual partner or using clean needles for people like drug users and so on. HIV can be treated using antiretroviral drugs or medications that target the different pathways of viral infection, viral replication, as well as the assembly of the viral particles. So these multiple drug therapies will stimulate the immune system, making AIDS kind of manageable over longer periods of time. So this brings us to the end of topic 6A. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.